Good morning, guys. I'm Amit Segamapatra, a junior research intern in the University of Milan, specialized particularly in the molecular biology of the cell. And if you have any doubts after this lecture, you can always provide me questions or any kind of feedback in the given mail ID below. So today I'll brief about the trained immunity and its role in health and disease. Sometimes I may be too fast to go through the slides, so you can always watch this video at a reduced speed, but yes, that is up to you and how you feel. So let's start. So we must wonder that we have heard about this innate immune system, the adaptive immune system, but what is this trained immunity and how does it get differentiated from the innate and adaptive immune system cells? Because we already know that the innate immunity is linked to the adaptive immune system. We know when in our body the innate immune system actually fails, the innate immune system gets triggered, or partially they both play a major role in killing a particular infectious disease, but disease related pathogens. So if we go through the definition of what a trained immunity is, we can easily understand that it actually describes the long-term functional reprogramming of our innate immune system cell. What is this long-term functional reprogramming? Reprogramming means activating again and again. So when the first time you will encountered by a disease, by disease causing pathogens, they will respond slow. The next time they will respond better. The third time they will respond even more better. So we always know that we are already concerned. We are always concerned about the trained immune system of our adaptive immune system cells, the memory T cells, the memory B cells. But actually, there is actually a trained immune system of our innate immune system cells, be it a macrophage, be it a NK cells, and that is recently published in some of the journals. You can go through in the ResearchGate or the NCBI. So I'll not brief about that, but I'll provide you some links so that you can go and understand. But here the trained immune system actually describes the long-term functional reprogramming of our innate immune system cells. Obviously, it may be evoked by an exogenous or an endogenous pathogens. We know that. And it actually leads to an altered response toward a second challenge. An altered response, obviously, because the response what we will get first time will definitely get better in the second time if you are not an immunocompressed or immunodeficient person. And for example, I can always say that that if we, as in our childhood we get injected the BCG vaccine, the Bacillus calmette-guerin. This BCG vaccine actually leads to reduction in childhood mortality, which is absolutely caused by some unrelated infectious disease. They not only triggers to help us to save us from the tuberculosis, but also some unrelated infectious agents. Now going through the relationship, as you all know that when exogenous pathogens gets inserted in our immune system, the pathogen contains the PAMP or the pathogen associated molecular pattern, and there is always a pattern against the receptor of the PRR in our immune system cells that actually triggers our innate immune system, and the immune system cells actually warns and serves a notice to our health by doing some kind of inflammation, some antimicrobial activity, or by releasing some kind of cytokines, and ultimately they actually kills and chops the antigen and presents some parts of it to the adaptive immune system cells, which is most likely to a T helper cells. So what I want to mean with this slide is that our immune system gets alerted every time when such kind of pathogen invades, and that's how our immune system actually gets trained. Here we take an example of the invading pathogens. For example, here is a Mycosporidia, which is an invading pathogens, an exogenous invading pathogens, which actually triggers the macrophage to get activated. The macrophage is there activated, and they actually kills and chops some part of the macrophage, and they also present it to our adaptive immune system cells. Not only this, the macrophage also play a crucial role in order to activate the NK cells by releasing some kind of cytokines. For example, here is interleukin 12. And in reference to that, the NK cells also release interferon gamma, which is actually helps in killing of the pathogens fully when the cell when the pathogens is actually getting recognized by the cytotoxic T lymphocyte. So what I try to mean from here is that this interleukin 12 is actually also helps in activating of the precursors of the Th1 and Th2 cells, which is the Th null or the Th0 cells. We know that the CD4 T subsets is actually the T helper cells, which actually corresponds with the APC of the antigen cells. For example, here is the macrophage, or in some cases there is some monocytes. The T helper cells release interleukin 21, and the Th and the APC actually release the interleukin 12. So whatever in this case, so whenever they gets recognized, the Th1 actually helps the Th1 actually activates the cytotoxic T lymphocytes by triggering the interleukin 2. And with the help of this interleukin 2, interferon gamma, interleukin 12, the cytotoxic T lymphocyte actually getting activated and helps in killing of these pathogens fully by releasing some kind of perforin or some granzymes and etc. etc. But in the Th2 cells, there is some unlikely event like what happened in the Th1 cells, because the Th2 cells actually activates the B cells, which actually forms the immunoglobulin, and when we know that by forming this immunoglobulin actually they fights with the invading pathogens and kills the invading pathogens. Let's go by the difference between the classic immunological memory and the trained immunity for innate immune system cells. So like us, we learn, we memorize, and that's how we get trained. But the immune system cells also, when is getting invaded by a pathogen, some part of the pathogen is decided inside the memory cell or the B cells, which is formed by the clonal expansion when the infection actually occurs. Now you must wonder, what is this clonal expansion? Clonal expansion is nothing. Whenever there is an infection, the T cell B cell fights for it, and if they are successful, or even if they are not successful in some cases, they actually keeps some parts of these invading pathogens, and they proliferate and make some clonal expansion, and some part of the T cells and B cells actually transformed in the memory T cells and memory B cells, and they get stored in our adaptive immune systems. Whenever there is a second attack of that invading pathogens, they actually comes first and fight with the pathogens. How? By making the specific T cell clone for the memory T cells and specific antibody formation for the memory B cells, because we know that the T cells and the functioning of the T cell and B cells is different. So the T cells makes the specific T cell clone, the T cell receptor clones, and the memory B cells actually forms the antibody, and in that antibody there is some specific amino acidic sequence which actually matches with the sequence of our invading pathogens, and that's how they get recognized with each other. But in the case of trained immunity of our innate immune system cells, which is an adaptive characteristic of our innate immune system cells, we cannot say it as an adaptive immune system. Remember, we can always say it as an adaptive characteristic of our innate immune system cells, because in the first infection, the strength of our immune system is always low because of the specific or unspecific nature of the pathogens. That doesn't matter. But in comparison of the second infection, it will take some more time, but this time with a better and enhanced client response. So how does it work actually? How does why our innate immune system cells actually not be functional, is not so much functional in the first infection, or how they gain function in the second infection? We'll discuss in our next slides. For example, here is an example of our innate immune system cells. The innate immune system cells, we know it's getting formed by our bone marrow progenitors. Now, if we inject some kind of stimulus in our immune system, for example, here is a stimulus. So when it gets injected, our bone marrow progenitors actually started developing the innate immune system cells. But that innate immune system cells actually goes some epigenetic reprogramming and metabolic adaptations, which is actually the reason for how they actually get trained. Now, these trained cells obviously have some good and bad effects. We'll discuss about this in the later slides. But obviously there is another way of making this innate immune system cells, not only by this training stimulus. We heard about the term clonal hematopoiesis. We know that during the clonal hematopoiesis, hematopoietic stem cells actually start developing into the different type of blood cells, which actually then start making cells with the same genetic mutations. Now, if there is a kind of mutations, then sometimes it can happen that your innate immune system cells getting overtrained or getting less trained. Why? Because of this clonal hematopoiesis. Now we know that there is some good and bad effects of everything, but if your immune system cells gets overtrained, then obviously you can encounter some disease such as autoimmunity. In further cases, if you need some kinds of transplantation, then you may face this transplantation rejection, and obviously your immune system will lead into some chronic inflammation as well. But the good thing is that you will get cross protection against all kind of infection, against all kind of diseases, if your innate immune system cells are particularly trained. So what happens? We actually inject some kind of vaccination, Let's suppose in our innate immune system cells, and for example, the macrophages are getting activated, and they undergo some epigenetic reprogramming in our innate immune system cells by doing what? By doing some kind of histone modification, DNA methylation, modulation of our miRNA. miRNA means the microRNA, and the long non-coding of our RNA expression. Now, all this is not important as much as the DNA methylation and the histone modification, because these are the main two factors that we are going to cover and discuss. So what happens after this epigenetic reprogramming? After this epigenetic reprogramming, actually our innate immune system cells getting trained, and they gain some transcriptional and functional programs, which leads into, I mentioned this already, the positive and negative effects, be it adaptive or the maladaptive. I'll discuss about this maladaptive states because actually we know that the epigenetic reprogramming occurs at stages when the developmental potency of cells actually changed. Okay, so in the case of maladaptive stages, the immune system goes into one drive, particularly in the disease of sepsis, because they release some chemicals into the blood to combat the infection, which actually triggers the widespread inflammation, but it can ravage our entire body. So whenever we are getting injected by a particular disease causing vaccine, there is always some risk of our innate immune system cells to get overtrained, but they're used in certain quantity so that our innate immune system cells can work perfectly. So for example, by using some kind of training agents, we can say for the BCG vaccination or the beta glucan supplements could actually strengthen the innate immune system actually to mount and obviously an increased innate immune response. For example, if we have some infection cause such as SARS-CoV-2. Now let's discuss about the different forms of the innate immunological memory. Now, as you know that whenever the monocytes of the macrophage is in resting condition, if they receive the first stimulus, they start raising the cytokines and they get activated. Now, when they get activated, they actually develop some kind of memory. Remember, I told you during the memory B cell and T cell development, they do the clonal hypothesis, and by this clonal hypothesis, some of the memory B cells and T cells actually transcribed into some kinds of these T cells and B cells actually gets transcribed into the memory B cells and the T cells. But here, these monocytes actually undergo some epigenetic changes. Some monocytes, some some monocytes, and some monocytes actually develop some kind of specific memory through this paired immunoglobulin like receptor. For example, here is a PIRA, which is very very specific to antigens, and there's specific to antigens that is being presented by the major histocompatibility complex 1, which is the MHC1. So as if they now receive the second stimulus, they will definitely get a better memory response and the release of better and high levels of cytokines. There will be releasing of the high cytokines, as they are now trained monocytes. But obviously in the case of PIRA, the level of the cytokines will be low if they are unspecific. What I want to mean is that, like memory B cells and T cells, which is very very specific, these PIRA paired these monocytes is also very very specific to that particular disease. Means, for example, if you get triggered by tuberculosis in these during a first stimulus, if you get triggered by the tuberculosis, means this PIRA will be very specific to tuberculosis. But the monocytes that goes the epigenetic changes will also encounter some other diseases, not only the tuberculosis. Okay, so that's how they develop a better memory response. Now let's discuss about some odd topics here. Not odd, it's definitely related to our trained immune system cells. Here's the mechanism of the in vitro BCG trends. It is a classic example of how our trained immune system actually works. Because when a naive monocyte cells are stimulated with the BCG, it's actually rests in some days. Then the monocytes actually engulfs and do phagocytosomal activity, which happens in the which actually helps in the release of the muramyl dipeptide. This is MDP, the muramyl dipeptide, along with the metabolic shift, which actually is generated by the AKT or the mTOR pathway. It is a protein kinase B, obviously. To the guys who don't know what is actually mTOR pathway, it is the mammalian target of the rapamycin. Here the MDP once binds with the NOD2. Sorry, here the MDP once binds with the NOD2, which is a nucleotide binding oligomerization domain. Sorry, the full name of the NOD2 is the nucleotide binding oligomerization domain containing protein 2. Okay, so you don't need to remember it. You just focus on the NOD2 protein, because it induced the epigenetic histone modification. With all this binding, they actually induce the epigenetic histone modification. Now this EAGM actually results in the increased access to the promoter regions of the genes that are related to the inflammatory pathway. So they actually targets the promoter regions of the genes to obviously recognize the better to recognize more PAMP of the bacteria, to more pathogen associated molecular pattern of the bacteria, because that's how they actually get trained. They can't get trained like we train a dog or a cat. They need some kind of development in their promoter regions, or in their upstream or downstream enhancer regions. So what this protein actually helps, they actually helps in changing these these histones modification actually. And as these monocytes are getting trained, they express high levels of pattern recognition receptors, and will also produce more cytokines if there is an invasion of any kind of pathogens. they will always trigger some kind of increased interleukin 1 beta, interleukin 6, or the tumor necrosis factor production, because they now are very very because they are now not specific to any kind of any kind of PAM. In fact they kind of recognize all kind of PAMs, because they now understand that this is an infectious agent, and we need to encounter it. So that's how actually monocytes get trained. They are always in the first line defense of our innate immune system cells. Throughout this graph, you can understand that the BCG training during the first encounter is very, very low. Why? Because they are untrained monocytes. They don't know what to do. But whenever they are in resting condition, they undergoes this epigenetic modification, and the next time when there is an encounter, they actually fight, and they show some increased level of the increased production levels of cytokines, interleukin, and all kind of pro-inflammatory cascades. So let's go to the next slides. If we understand about what is actually trained immunity in humans, because it is a very complicated method, but it works actually very well. For example, a vaccine of smallpox will not only save you from the targeted disease, but also from other infected disease, because that's what actually vaccines do. For example, here we discussed about the malaria causing infectious agent, and we actually nurtured it into mice. So what happens? The mice is getting infected. And the Plasmodium infection is actually sufficient to promote the MyD88 mediated caspase 1 activation. This MyD88 caspase 1 activation is very, very important in order to activate all these proteins. For example, it's very, very dependent on the interferon gamma priming. Because the expression of this NLRP3, the expression of the ASC, the expression of the P2X7R, of the NLRP12, all these actually are a kind of inflammasome components. So these inflammasome components actually helps in triggering our innate immune system cells to not cause other disease when you are being encountered by a Plasmodium vivax or any kind of disease caused by malaria. Now, as a consequence of this Plasmodium-induced caspase 1 activation, the mice produce extremely high levels of interleukin-1 beta upon second microbial stimulus, because it can become very, very hypersensitive to any kind of septic shock. High levels of interleukin-1 beta, we know that if there are production of the high level of the interleukin-1 beta actually, upon a second microbial stimulus, means if there are invasion of another pathogens when you are encountering malaria, the mice will become hypersensitive to septic shock. So from this we can conclude that NLRP12, NLRP3 dependent activation of the caspase 1 is likely to be a key event in mediating the systemic production of the interleukin-1 beta and the hypersensitivity to secondary bacterial infection during the malaria, any kind of secondary bacterial infection during malaria. Now let's discuss about the cell components which are actually getting trained. If you discuss about the central trained immunity and the peripheral trained immunity, is that there is some invasion of the vaccine or the adjuvant in our innate immune system. For example, BCG or beta glucan, they develop into the hematopoietic stem cells. I mean they are developed in the hematopoietic stem cells, then they transform into multipotent progenitor cells, to common myeloid progenitor cells, to granulocyte monocyte progenitor cells, and then they develop into monocytes and come towards the peripheral immune system. So by using these training agents like the BCG vaccine or the beta glucan supplements could actually strengthen our innate immune system to mount an increased immune response. For example, if we are getting encountered by a SARS-CoV-2 vaccine, SARS-CoV-2 infection. And what happens in the case of this peripheral trained immunity is that the epithelial cells actually gets trained to store the inflammatory memory. And that's how they train macrophages, the NK cells, and the monocytes to equip them more powerfully, with the ability to mobilize maybe more fast during if there are any subsequent assaults, or there are any invasion of the second pathogens. Now you will get surprised to know that even NK cells, which is a part of our innate immune system cells, which are actually part of our immune system cells and play a very crucial role in our first line defense. They also actually possess some adaptive immune characteristic, and if you go through some articles, I will provide link of the thesis article, that how NK cells also works as memory NK cells, and they also play a crucial role in our trained immune system of our innate immune system, because they possess the adaptive immune characteristic if they get infected by the human cytomegalovirus. For example, if a woman encounters cytomegalovirus during the first pregnancy, the NK cells will develop a better response if she encounters this disease in second pregnancy as well, because they're capable of making memory NK cells and undergo expansions and can more fastly release cytokines and actually destroy them. So by this characteristic, we can also identify the nature of the natural killer cells, and obviously they undergo some modifications, such as the monocytes and the macrophages go. Now, now we are all discussing about this epigenetic mechanism, how they actually undergo this kind of development. Now, as I mentioned, if the monocyte is in unstimulated, or in the steady state, we know that the chromatin is in highly condensed state. They show high DNA methylation, and the expression of the genes is very little. But whenever they are getting triggered by an invading pathogens, they release cytokine. So not in the manner like a trained monocyte would do, but obviously they release cytokines, and the chromatin of that highly condensed DNA is now is now starting to open, and they show low DNA methylation, and also there is some active transcription of the H3K27AC and H3K4ME3 genes. Now this modification happens to H3K27AC to the DNA packaging protein histone H3, which actually works as an active enhancer mark, and also plays a crucial role in the activation of the higher activation of some transcription activities. Now, in the case of the H3K4ME3, which actually promotes the gene activation and also try to remodel the chromatin. So these genes are actually help very much in developing the epigenetic mechanism in the monocytes or the macrophages. Because activation of these genes actually means that they make the DNA in the chromatin accessible for the transcription factors, and we know that if they start making this transcription factor more, they start developing promoters and enhancer, which actually recruit the PAMP, and can function more openly to encounter any kind of disease that we are facing in our immune system. Now let's understand about this trained and unchallenged condition monocyte. If the monocyte is in resting conditions, means they are getting trained. Although they are unchallenged, they are getting trained. So here we can see that the chromatin is in mildly condensed state, not as much condensed as it was in the steady state, and the methylation in the DNA is also low, and there will be only activation of some little genes such as H3K4ME1. But when they are getting restimulated again, they are getting trained. When they are getting trained and challenged again, they actually show a better production of the cytokines. They actually show an enhanced production of the cytokines, and also the chromatin of the DNA is starting to get opened again, and they express all kind of genes, be it H3K4ME1 or H3K4ME3 or H3K27SE, and they also show low DNA methylation. And as I already mentioned, that these modifications happens to H3K27SE to the DNA packaging protein histone 3, which actually works as an active enhancer mark, and they actually helps in higher activation of the transcription factor. And for the case of H3K4ME3, they actually works as a promoter in the gene activation, and they remodel the chromatin structure, which makes the DNA in the chromatin accessible for the transcription factor. So that's how the epigenetic mechanism actually works in our naive monocytes to become a trained monocytes. So we are discussing all day along with our innate trained cell. So does it mean that only one dose of this of any kind of vaccine or the adjuvants is enough to trigger our memory cells to memorize and to form, and from then such virus or pathogens cannot attack our immune system again? The answer is no, because it varies among individuals, and when there is a pandemic, it's always to be in the safe side by getting on two more booster doses to keep yourself always safe. So what happens when you take a dose of COVID vaccine is the spike protein in this vaccine is actually which actually help to get recognized by our innate immune system and by adaptive immune system, because here helper T cells actually detect the spike protein and alert our immune system cells. And we all know that the process of the killing of the killer T lymphocytes, the process of killing the COVID virus by this killer T lymphocytes, they actually secret for this spike protein, and B cells, we know that they create some specific antibodies to specifically recognize the spike proteins. So these T and B cells actually develop our immune memory of this spike protein, which actually contains the COVID virus, and whenever there will be a next encounter or recounter, they actually start seeking for them again, and will kill them in advance. And these antibodies actually in the body, in our body, kept for some times, because they are not always permanent, but you can be sure safe for some years, because it actually varies from individuals to individuals. For example, why we are advised to take a second vaccine dose, because we know that months later or years later, our immune system may gets compromised. So in order to train our immune system, if we take a next dose or a more or a kind of booster dose, this actually strengthens the response and creates an immune memory to the virus. So whenever our immune system is triggered by the SARS-CoV-2 again, if they encounters this virus in future, they actually prevents infection and some serious illness. Obviously, you might face some kind of dizziness or kind of symptoms, or kind of some symptoms, but you'll be well protected, because you know that your memory T cells and B cells actually functioning well. But here there is the role of the memory T cells and B cells. But what is the role of our innate immune system cells here? So to discuss that, I can suggest you to go through some recently published research articles, that I'll provide link in the description below. And at the same time, so what is the role of our innate immune system here or more precisely our innate immune cells here, to get rid of COVID disease for the second time? The answer is, like the T cells and B cells starts to memorize, our NK cells, the monocytes, they also memorize, as we discussed in this whole part. They actually also helps the memory B cells and T cells in order to in order to remove the effectiveness of the spike protein if it's getting encountered again. Now, as we know that the vaccines have some traditional approach. The memory T cells, B cells, they are getting formed. They actually kill the infected cells. They create antibody. The memory T cells create antibody, and they have the specific amino acid sequence to target the pathogens when they are encountered again. But for the trained immune system, they actually don't have some specificity. The main difference between the adaptive immune system cells memory and the innate immune system cells memory is that the specificity. The adaptive immune system cells is very specific. Our innate trained immune system is very, very unspecific, because if they are getting trained, they will start killing any sorts of pathogens. They will not recognize the specificity of such embedding in cells, and they will always show an enhanced responsiveness and protection. And that is why our innate immune system cells is rapid and robust. We use the term rapid and robust because our adaptive immune system cells, even the memory T cells and B cells, will take some time to recognize the invading pathogens, but our monocytes and neutrophils will not. So here are some good and bad effects of the trained immune system cells. As you already mentioned, that if they are getting excessive trained, they are getting overtrained, then you may encounter some kind of autoimmune disorders, for example multiple sclerosis, or a kind of organ rejection in your excessive trained immune system, and also some kind of inflammatory diseases, for example allergy and cardiovascular complication you may face. But the good thing is that if someone who is immune tolerant, they will not face this kind of diseases. If our innate immune system cells are properly trained, then there is no chance of getting diseases, and in fact our immune system will perform carefully and very, very constantly. So that's it. I think that's the end of the slide. Yes, thank you guys for being part of this lecture, and I hope